Mubareka. I'm an infectious diseases physician at Sunnybrook Health Sciences Centre and a virologist at Sunnybrook Research Institute and the University of Toronto. We're really kind of starting from scratch when it comes to this coronavirus. Now, having said that, this is not the first coronavirus that we have seen. So there are 4 coronaviruses that can infect humans. These are common cold viruses that surely almost every Canadian has been infected with. So we're all coronavirus experienced in one way or another. And now there are 3 what we call high consequence coronaviruses. So those include SARS, MERS, and now SARS coronavirus 2. So of all of those coronaviruses, there are very few that have been extensively studied, at least in Canada. There are a couple of centers of excellence where studying things like MERS has been a main focus. So I'm referring to places like Vito Intervac, where they've done some excellent work on MERS coronavirus. But having said that, a lot of the virologists in Canada were mostly working on other viruses when this hit. So one thing that this pandemic has done has galvanized that community, and many of these virologists, whether they were working on HIV or let's say Zika virus or West Nile or influenza, pivoted over to coronavirus because there are a lot of basic skills that can be applied. And again, across all those different fields I mentioned before, whether it's epidemiology or basic science or clinical research. So that's one thing that has happened. The other thing that has happened really is across the broader research community. So there have been individuals who have come forward who have, long-standing reputations and very storied careers in other fields. It might be oncology, it might be neurosciences, it might be engineering. It could be really any field that's related to biology or even sometimes in terms of physical or physics. Those are all things that can be brought back to try and understand this particular virus. So I have met more investigators in other fields in the last perhaps 6 weeks than I have throughout my entire career, and it's been great. I think where we can do better is really trying to coordinate these efforts. As you probably appreciate, the way science works in Canada, there is somewhat of a competitive atmosphere. And that's partly because there are limited funds, and these are, to gain access to these funds, that there are competitions. And likewise, even on the other side of things, once there are results, in order to publish, in some fields, there can be a race to publish certain results. So that can lead to an atmosphere where people remain a little bit closed. They hold their cards close to their chest because they don't want to give away they don't want to give away too much. And I think that that's what has really changed with this particular pandemic. The spirit has really changed. I think individuals have really opened up. I have heard the phrase repeated many, many times by several people. It's better to collaborate than compete, which is a huge relief because I don't think that it would really help anybody to maintain a competitive attitude in this particular situation. So that's very helpful. So at least I think attitudes have changed and opened up. But again, I think what we've been lacking is more of a coordinated effort. Part of it is because many of us don't know each other, especially across different disciplines, for example. So I'm referring, just as an example, perhaps many of the engineers and the virologists do not normally work together, and now there are very clear opportunities and needs for them to work together. But if that kind of approach could be coordinated, I think we'd be further ahead. The key thing about isolating the virus is it was a tool, a very important tool that we now have to develop a number of medical countermeasures. And that really starts with understanding the biology of the virus. So unless you have the virus in hand that you can use to infect cells and other models, it's kind of difficult to understand what the potential druggable targets are, how the virus affects the host, whether it's just a cell or an actual organism. So now that we have the virus to be able to do that, that's incredibly helpful. And this is not something that we are doing alone. There are a lot of collaborators, both at the University of Toronto and other institutions that we're working with, who have potential candidates as antivirals or who have potential candidates as vaccines. So with the virus, we're able to actually test those candidates. Without it, we wouldn't be able to even consider doing that. To begin with, it was quite remarkable how quickly the first tests were developed. So these primers, they're called the Corman primers, were based on the very, very early genomic sequences that were posted from China. So that was incredibly helpful because you can't develop a detection test if you don't know what the sequence of the virus is. So it was very helpful to develop a PCR test. So that's a type of molecular test that is very commonly used for respiratory viruses. So that's helpful. Again, using a lot of the infrastructure we already had for things like influenza, doing detection for those other respiratory viruses that circulate every year. At least now, I won't say it was as simple as a plug and play, but we were able to implement tests that were more specific for SARS coronavirus 2, but using a lot of the same platforms. So that was incredibly helpful. We can definitely do better. We know that we've had issues with supply of different reagents, also supply with swabs. And also there's what we call a turnaround time. So someone might have a sample taken, and depending on where you're getting tested, it might take a day, it might take a week to actually get results back. So there are a couple of improvements that we're making from that perspective. And again, when I say we, obviously not just our group, but I'm talking about the broader scientific community here. So developing point of care tests is really key. So these are tests where you don't necessarily have to send the sample back to the lab. They're more portable, so you might even be able to do the testing right away and get results within an hour or two. Also being able to try and take a different type of sample. So being able to take something like spit or saliva as opposed to a swab. Now, we are looking to see whether that's better or not. It'll be very, very important to make sure that whatever we do is not inferior to the current testing modalities, so that's key. The other type of test that is important to develop, and people have been making good headway on this, is really the serological testing, because understanding whether someone actually has antibodies, so this isn't so much a detection test, but a test that tells you whether or not you've been potentially exposed. Now, not everybody might make antibodies. We don't know a lot about who does, who doesn't, how long they last. There are a lot of unknowns there, but it all starts with having a really good test for that. So these are the advances that people are really focusing on right now from a diagnostic perspective. In terms of really understanding what the priorities are in terms of research and especially medical countermeasures, it all starts with understanding the biology of this virus. So the virus is relatively similar to SARS coronavirus 1, but there are obviously what we call phenotypic differences. So the disease that it causes in humans is somewhat different. This virus seems to transmit better. And also, we hope, and it looks like that's likely, has lower mortality than something like SARS, coronavirus 1. It certainly has lower mortality than MERS, very fortunately, because the mortality for MERS is quite a bit higher. But understanding why that is really important. So understanding the biology of the virus itself, what it's doing in cells, which cellular proteins it's interacting with, which cellular proteins it's stimulating, and which other host cells is it stimulating. So in other words, trying to understand the host immune response to it. Those are all really key priorities. Also, or mainly because by understanding that biology, you'll understand what the druggable targets are. And if you understand what the druggable targets are, that really helps people develop screens for possible antiviral candidates. And it also helps people develop or design, it's called rational design for antivirals, design molecules that can target either components of the virus or components of the host response that would mitigate infection. So that's why it's so important to understand the biology. The reason it's also important to understand the host response is it has an immense bearing on vaccine development, and also understanding what are the correlates of protection. Because we'll only be able to really test vaccines if we really understand what the correlates of protection are. So those are a couple of the key priorities I think that in the coming months will be highlighted. Genomics play a really crucial role, and it plays a very so the perspective is actually quite interesting because you can look at it from the most minute cellular level. The genomics at the cellular level are very, very insightful, but because by looking at how the viral components of the virus actually interact with the components of the cell, kind of goes back to what I was saying earlier about identifying some potential druggable targets and means by which to interfere with that host viral relationship. So looking at genomics from that perspective is really, really key. And obviously it goes beyond the gene. It can go to the transcriptome and the metabolome and so on and so forth. So there are quite a few systems, and we call this systems biology in terms of understanding what's happening at the cellular level, particularly when a host cell and a virus interact. But then there's this whole other level, the population level, which is really, really important. And again, there are two sides to that story as well. So there's the host side of the story, and then there's the viral side of the story. And both can be sequenced, and we can get the genomic signature from both perspectives. So humans are obviously incredibly diverse in some respects in terms of our genome. And I think there's a very good chance that if we do this properly and in a broad enough fashion and in an organized and coordinated enough fashion, we might be able to answer some really key questions. So for example, we know that some people do poorly because of their age, because of comorbidities. But we have seen some elderly people do well, and we've also seen some young people do poorly. So what is it about those two different hosts that make them that determines their outcome essentially? So understanding the host genome in the context of viral infection and outcome or severity of disease will be really, really important. Then there's the viral side of the story. So if we understand the entire, or if we are aware of the entire viral sequence and over a range of different viruses, and here we're talking about thousands, tens of thousands, perhaps even hundreds of thousands of viral sequences across the globe, then we'll be able to make some really important links and understand the relationship between those viruses. We are hoping, of course, until we have the data, it'll be difficult to say with certainty, but at this point it looks very promising that we'll gain some significant insight into viral activity and transmission if we do in-depth enough analysis of the genome and broad enough analysis of the genome over time and over geographic jurisdictions. The other thing that the viral genome can tell us, and I know I keep coming back to this, but it is important, is looking at components of the virus that are important in terms of vaccine response. So there are parts of the virus called viral epitopes that if we understand what the viral epitopes are for, so these are the targets for the immune response that we're trying to solicit by using a vaccine. If we understand what those are, that's a great starting point. But as vaccine trials roll out, as vaccines actually get rolled out at a population level, chances are those epitopes will change as the virus passes from one person to another. So genomics will be really key in terms of tracking that change and doing surveillance in terms of anticipating vaccine escape mutants. And similarly for antivirals, as people start using antivirals, there's a risk of antiviral resistance. So if we have the viral genome at our fingertips, hopefully we can anticipate some of that and intervene. And lastly, we really need to know the viral genome in terms of diagnostics. So I mentioned earlier that we target the viral diagnostics against the viral genome. But again, if there are changes in the genome, there's a risk that we'll lose our ability to detect the virus. So that's why it's important to do ongoing surveillance and just verify our primers and our targets to make sure that they will still detect the virus. In terms of data sharing, there's a significant role for genomics. I think that it'll be absolutely crucial that we share data across jurisdictions. If we are just, for example, looking at data within an institution, within a city, or within a province, there'll be really a limited amount of information that we can glean from that, and it'll be very difficult to make decisions based on that limited information. So if we want genomics to really impact decision making, there needs to be data sharing. Obviously, there are a number of very important considerations around privacy, and also just cost and infrastructure. How do you curate the data so that the data is consistent? It has to be quality data. But if we make those investments in that infrastructure, I think that that will be very, very high yield proposal. So in terms of preparedness, genomics stands to play a very, very important role. So right now, we are in the first wave of the pandemic. We anticipate further viral activity, whether it comes as a second wave or smaller outbursts. Genomics will be really, really important in terms of being able to try and track the virus through communities and populations, and maybe even help us with surveillance in anticipation of further waves. So it's quite key to have the information now rather than waiting to generate it later, because it'll only be useful if we can apply it to mitigate further spread. One of the ways that we can be better prepared, especially in terms of policy, is not so much to completely change the policies that are in existence, because we really have a good regulatory framework in Canada, and it's important in terms of doing this work in a safe and responsible manner. But it could be done more quickly. Now, I have to admit, having said that I've never seen anything so quick as what I've experienced with this in terms of research ethics and with legal. Things have moved incredibly quickly. But if we could do it even at a more accelerated pace, we would be better off. And I think the way to do that is to have pre-existing agreements. So I think one of the things that is important to realize is that there's been a lot of attention on funding around this particular pandemic. But this area has actually been chronically underfunded for a very, very long time. So basic science research in virology is something that's been quite limited in terms of funding support. But I think, again, we write this every time in our grants. You know, there will be an epidemic, possibly a pandemic, when this will become incredibly relevant. And unfortunately, we become very complacent in between pandemics. And we saw this with SARS. We've seen this with other viruses, including influenza virus after the 2009 pandemic. And I have to say, I think we are paying the price for that complacency. That may sound rather harsh, but I just want to underscore that, because if, again, in 12 months, though that's being optimistic, or 24 months, this is behind us. If we forget about the ongoing need for research, especially during interpandemic periods, we will find ourselves in this situation again. In terms of the silver lining, I think what has heartened me, and to be honest, what has kept me going, has been the fact that there has been so much openness and collaboration. I've just not seen people rally together like this ever. So I think if we continue with that momentum, we will be able to make a significant difference. I don't want there at the end of this for us to lose that particular momentum, because the collaboration has been incredibly productive. Now, admittedly, a number of individuals will have to go back to their day jobs and continue to do the good work that they were already doing before this pandemic. But having said that, there's no reason that we can't capitalize on and expand on the collaborations that have now been established. And again, I'm really talking about some of the collaborations between physical sciences, engineering, virology, biology. You know, the amount of innovation that has come through in the last month even. Just to give you an example, there are individuals at our institution I had no idea, they're physicians that I had no idea were engineers. And you know, they've applied a lot of the skills that they learned through engineering to innovate new solutions, because they are at the front lines and they know what's needed. And they've been able to turn to their colleagues and apply their own skills to really close a few very key gaps that we've witnessed. So I think, again, if we've galvanized, we need to consolidate that. So that's one of the silver linings, I would say, that has come of this. And there's no reason that we can't continue to work together in the same fashion. Much in the same way that there's no reason we can't continue to be as kind and supportive of each other as we have been during this. And I do think that there will be a long-lasting effect. The other silver lining that perhaps is less related to to humans per se that I've noticed is the fact that, with decreased human activity, there's been a pause. And we've seen some rejuvenation of the natural world. And I think that for me, certainly, it's made me rethink about how I actually impact the natural world and my daily activities. If in a way, we can almost use some of what we've done. We've actually adapted quite well in terms of having meetings virtually. Of course, face-to-face meetings are always great, but if we can mitigate in some way travel and the actual impact that we're having on the natural world while we continue to do the activities that are necessary both economically and for healthcare and for research, I don't see why we can't carry those things forward either. In terms of an interesting perspective on this virus, I mean, this small, tiny little molecular machine has very effectively brought the world to its knees, essentially. And to me, that's just a very fascinating concept. To me, this virus is not necessarily the problem. It is a natural phenomena that was very quietly residing in its natural habitat, most likely a bat. And really, it is human activity that caused this entire pandemic. I certainly don't want to lay the blame at the feet of the individuals who have been infected by this virus. That is by no means what I'm trying to imply. But what I do want to point out is the bigger picture of this. So as we encroach on natural habitats, we increase the likelihood that we will encounter or be exposed to viruses that normally we would not be exposed to. So that's one thing. So that's the spillover event. But then because of global travel and pretty much unfettered global travel, we very effectively spread this virus around the world in a very short time period. We need to think about how our behavior really allowed some of this to happen. Because again, if we want to mitigate this in future, we have to take some responsibility and accountability. I'm Dr. Samira Mubareka, and this is my Future Economy.